All right, here we go. The reproductive system. The reproductive system is one that's interesting. It's the 11th of our 12 systems, and it's unique in a couple of ways. Number one, rather than being similar in its function in males and females, it actually divides humans into males and females. And that division represents their distinct role in reproduction. So it differentiates our bodies into one or the other. The other thing that's interesting about this organ system is that you can actually remove it and live a healthy life. Neutering does not kill, whereas removing any of the other organ systems would kill you. Despite that removable aspect, it does have a profound effect on differentiating our bodies that run the whole gamut of human metabolic reactions. So not only does it differentiate the appearance of our bodies, it has a profound effect on the ultimate size of our bodies, as moderated by our own genetics. But it also it also affects our behavior so that we fulfill a certain role and recognize the other sex for having the other role. We are going to see a system where the organs that we'll describe today produce a special set of cells that go through the second kind of cell division, meiosis, in order to achieve a gamete. A gamete is an exact half set of the chromosomes and genes in the cell of the parent body. Now that exact half set must unite with another exact half set in order to bring the chromosome number back up to 46 and the gene complement to a diploid or duplicated complement. So in order for reproduction, the contribution of two individuals in reproduction to work, you're going to have to cut the genes in the cell in half in a very precise way and then bring them back together in a precise way in order for the genes to remain balanced and for the genome to remain continuous. Female system, just to give you an overview, is important because it's regulated by sex hormones and produces functional gametes, but also the female body protects and supports the fertilized egg through embryology and fetal development, what we call gestation, until it's ready to separate from the mother and be born alive. But it also, after birth, it supports an extremely long period of growth and development. The newborn infant is essentially helpless. The thing to remember about the female system is it's emphasizing high quality. The entire female body goes through a 28-day cycle to release usually one egg. Now that egg is unusual. It's massive. It carries the organelles that will be present in the offspring. It carries nutrients to get it through those initial rounds of cell division. And in fact, it is comparatively limited. Although the ovaries of the female when she's born will carry thousands of potential eggs. They're already biochemically defined at birth. Despite that fact, if you look at the idea of, for instance, 28-day cycles, that's about 12 cycles a year. And if you're really generous and you don't have any pregnancies to stop ovulation, if it's 40 years of fertility, that would be 480 eggs. 50 years would be 600 eggs. So although thousands are potentially there, the entire life of a female will produce at maximum 500 to 600 eggs. So limited number, but high quality every time. Now, the male system is stark in its contrast, going to produce gametes called spermatozoa in almost unimaginable numbers. The sex organs in both male and female are called gonads, and the male gonad is called a testis. The testes is where the gametes are produced. They are produced continuously after puberty and stored in the epididymis, where they are stored and protected. Because the sperm is so different from the body cell, it's a half set. The protection mechanisms of the body, including white blood cells, would see it as something different. So would attack it. Nurse cells are around to provide a kind of a region of biochemical protection so that the stored sperm are not attacked. After the behavior of mating is experienced, there's a period where the climax is reached and sperm are ejected through collected tubes and glands. On that trip outside the body, they are enriched by the materials from different glands to change the stream of sperm into semen and to eject them from the body. And that's kind of a summary of the biological requirement of male function. Now, the overall idea here is, remember, high quantity. We're going to reduce that cell in its division. In fact, we're going to keep things like mitochondria and membranes. We're going to keep a half nucleus, but other than that, the sperm just has a flagella, a motor. So it's very, very small compared to the size of the egg. The big contribution is that half nucleus, one set of 23 chromosomes that will complement the chromosomes in the egg and start you out, fertilized egg, with 46. So let's take a look at the male functions first. First, accessory organs. They're going to secrete fluids because it is necessary to supplement the sperm themselves to give them a good fighting chance to get to the egg. So along the way, we will see that the sperm duct is joined with several specific glands. Seminal glands are sometimes called seminal vesicles, add an amount of liquid, and they also add a charge of fructose. Fructose is a carbohydrate, high energy. Can be used directly by those mitochondria at the base of the flagella to make ATP. Because the whole role of the sperm is swim, 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 this is an important addition. The prostate gland now encloses that sperm duct. It's going to add some enzymes and anticoagulants. It turns out that when you put the sperm into the female reproductive tract, it tends to clot up, and these anticoagulants keep it liquid so that the sperm has something to swim in. Finally, the bulbourethral glands are going to inject some mucus that is alkaline in pH. And that's because the vagina is acid as part of its protection from invasion by pathogens. The male equipment includes a membranous, a skin bag called the scrotum that encloses the testes and holds them outside the body cavity. This must be important, and it relates to the temperature that you store the sperm under. It turns out 1 or 2 degrees makes a big difference in terms of sperm motility and sperm longevity. I say it may be important. How do I make that statement? Well, I make it from the fact that vertebrates who are mammals, the males all have a scrotum and an externally positioned set of testes. The sperm duct, after semen is formed, is going to move through a long term in the penis, and that tube is going to deliver the sperm outside the body. So we have seen this before when we were looking at the urinary tract in this first half. Definitely shows us, let me get my pointer moving. Pointer, there. This definitely shows us that the urinary bladder and the urethra up here, just a urinary tract, but after this junction, this Y shape, a urogenital tract. Now, in terms of daily use, of course, this is mainly this urethra. It is a tract for urine elimination. But during sexual episodes, it is going to be joined by the sperm flow from the testes where they are produced and the epididymis where they are stored through this long ductus deferens, a sperm tube that winds up around the bladder and is connected in the posterior part to the prostate gland. You'll notice that here is the base of that seminal gland or seminal vesicles. Here is the prostate, and here is the bulbourethral gland. As the sperm flows through here, it is supplemented with fluid, with fructose, with enzymes, anticoagulant, and eventually with alkaline mucus. on its trip out of the penis. This is just the second half of that, showing you the loop behind the ureter and the more complete drawing of the seminal gland leading into the prostate. On the development, the positioning initially of the testis is high up in the body cavity. 2 months shows this, but the connection of a gubernaculum testis tube down toward the position of the penis. As the body grows, so here's 3 months and down below 4 months, the relative position shifts as the testis descends through the body cavity to this position within the pelvic cavity and eventually descends into the scrotum. So that's a normal part of development. Recessive mutations can prevent the descent of a testis, and often that affects fertility of individuals that possess that trait. The spermatic cord is shown here, a braid of tubes of different kind. Of course, you need a rich blood supply. All that meiosis and the muscular contraction at sex has to have oxygen and nutrients. You also have to have innervation from the genitofemoral nerve right here. And then finally, this is again the ductus deferens, the sperm tube that's basically ascending from the scrotum and testis level up above the urinary bladder and around to the back for its joining to the other urethral tract. Temperature regulation, as I said, in the testes is quite important. Testes need to be kept a little lower than body temperature in order to maintain maximal sperm quality. This is, as I said, a kind of a universal property of mammal vertebrates. Muscles do relax and contract to kind of change the position of the scrotum. So in cold weather, muscles pull that up closer to the body, and in warm weather allow the scrotal sac to descend to maintain this acceptable range, or the most acceptable reachable range of temperature for the sperm. Now, reproduction means that we have to revisit cell division. And this is our first type of cell division. Mitosis is equational, and its products are all identical. In fact, I want to go back and take a developmental viewpoint. The fertilized egg that started your life had 46 chromosomes. Those 46 chromosomes and all the DNA and all the genes that they contain were copied exactly every time the cell divided. So when you went to 2, 4, 8, 16, you ended up with 16 cells that were exact genetic copies of the fertilized egg. And that continues up through the growth of your body. We talk about the reproductive system, and so we're going to talk about the gonads of the male and the gonads of the female. Those cells are diploid cells, exact copies. So mitosis, equational, and it accounts for growth throughout our lifetime. Within the gonads, there's going to be a different kind of cell division, and its only purpose is to produce gametes. It's called meiosis, and we'll get to that in just a bit. So what I want you to see in this diagram showing mitosis is the exact copying. Here's your starting cell with 6 chromosomes, and the colors indicate 2 sets. So there's a blue set, dark blue and light blue. There's a red set, dark red and pink, and a black set, dark black and gray. Now, what the sets actually mean, one of those, let's say the dark blue, came from the father, then the other one, the light blue, came from the mother, equal contributions of those chromosomes to make this set complete. Now, we're only showing 6 of the 46 chromosomes, but the result of mitosis is shown over here. This set is exactly copied in both the daughter cells, and that continues throughout your life. It's by DNA replication and chromosome replication, producing double strands of each of the types. And then this black doubled chromosome will separate. One strand will go one way and one another, so that each daughter cell has one black chromosome, just like you started with. So mitosis, equational growth. Meiosis, we're going to add an additional couple of processes that produce genetic variation between the products as they separate. In fact, these are two called segregation and assortment, and they are so effective in scrambling the genes that are carried on these chromosomes that functionally there's just not a limit to the number of different gametes that we might produce. This shows one option. Here are the chromosomes and we copy them, but this step shows the first thing that's different. Notice how the double black and the double gray, the sets, the number one chromosome that pair together, form this now four-stranded structure called a tetrad. This organizes the chromosomes in a different way so that when they divide, you can see that both blacks went this way and both grays went this way. So at the end of the first division, although you went through the same starting, you replicated, but then you paired, you separate those pairs, and what you end up with is kind of the same number of strands. Of the black-gray, you still have two strands. But notice all the black genes are over here and all the gray genes are over here. That's genetic difference. Now, the second division then separates these individual strands and halves the number. The thing you want to notice about these products, and this is only one outcome that comes from a particular kind of pairing and separation, but there are other ways you can separate the pairs from here to produce differences. What you always end up with is one of the black-gray, one of the red-pink, and one of the dark blue, light blue to make an exact half set of these. So if you call these 1, 2, and 3, you start out with 2 of each, you end up with 1 of each. That precision in the spermatogenesis and in the oogenesis means that when they unite, you're going to come back to that 2-set arrangement. That will be the normal complement for human reproduction. So over here, what do we notice? We start out with these same 6 chromosomes. And when we're down here, we end up with now 3 chromosomes, but not just any 3. You'll notice down here there's no gametes that have a black and a gray and a blue and no red. They always divide evenly if they end up as functional gametes. Now, with that being said, once you get this half set, there's then a cell development process. And in the male, it's called spermatogenesis, or the production of sperm. It involves a reduction of the cell. Organelles are thrown away. So you don't need Golgi apparatus. You don't need endoplasmic reticulum. You retain the membrane. You build a little special capsule here for digestive enzymes that are going to function when if it gets to the egg. You have the half set inside the head, and then you have this collar. This is where the flagellum attaches and all the mitochondria are packed in here. So the point is, make ATP, switch that flagellum back and forth, and swim, swim, swim. This process of maturation takes, as you can see, several weeks. And those sperm are produced with equal complements. So back here, we produced from each starting cell, we produced 4 gametes. Those gametes produced normally 4 sperm. So each one of the products of meiosis are viable in terms of sperm. And the other feature that's interesting is that many, many, many diploid cells go through meiosis in the human male. The human male is making tens to hundreds of millions of sperm per day, and there is a kind of a capacity. You know, the epididymis can only hold so many sperm. When it's full, that might slow down a little. But in response to a sexual episode, the body cranks up and starts making sperm immediately and quickly. So hundreds of millions of sperm are typical in the male body. Here's what a sperm looks like with its head, neck, and tail. This is the standard flagellum that we saw the microstructure of, just like flagella on microorganisms. The mitochondrial spiral organizes the mitochondria to produce ATP right where the motive force is needed to make that flagellum move back and forth. Now, with so many sperm produced and so many sperm used, tens to hundreds of millions of sperm will be deposited in a standard sexual congress. We do see an element of sperm quality. Is the sperm normally formed? Is the head and tail appropriate in its attachment? If you look at sperm samples, you will see abnormal sperm at times. Sperm where the head is sideways. I don't see any in this particular figure, but sperm, I have seen sperm where the tail is forked into two ends. Those abnormal sperm do not function. It's only the sperm that are complete and perfect in their development that have any chance, because the trip from the vagina up to the ovary is so rigorous that if you think about a typical sexual event depositing, let's say, 200 million sperm, if 50 sperm get to the ovary on each side, that's considered really good sperm performance. When we look at a real dissection, opening up scrotum, so you can see inside, here's the testis tissue, and it's encircling epididymis, a storage organ where the sperm are basically piled up, kept in a storage tank, and protected from the male's white blood cells. When sex is completed and climax occurs, the sperm are ejected up through one of these tubes of the spermatic cord. In this case, this is the ductus deferens, and basically up on that trip around the bladder and into the glandular region. When we see this overall, it's quite a trip from here all the way back up behind the bladder, and behind you can see it's going to pass by those openings from the ureters above that are filling the bladder, and behind, coming down into the prostate, they're separate. But right down here is where the ejaculatory duct joins that sperm path to the urine path. So from here down in the prostate gland, we have a urogenital tract. As it proceeds down, this is where the openings from the different glands, the seminal and the prostate, and down here the bulbourethral, or the bulbourethral gland openings, are changing that stream of sperm in just some body fluid into semen, which is specifically adapted to perform its fertilization task inside the body of the female. Now, this is a organ that is flaccid under normal non-excited conditions, but it contains these cavities of porous tissue, the corpus spongiosum and the corpus cavernosa, which are emptied of blood under, as I said, non-excited conditions, but when it's excited, blood flow is directed into these, making the penis erect, and then improving the chances for insertion and a deposition of sperm deep inside the female's reproductive tract. How do we control these processes of gamete production and preparation? And the answer is the same in both sexes: hormones. This is a major role of the endocrine system, and they begin in the brain, behind the blood-brain barrier, with the hypothalamus and pituitary. Gonadotropin-releasing hormone is synthesized in the hypothalamus. It is a function of that junction between the brain and the endocrine system to make the gonadotropic-releasing hormone, which is then carried to the pituitary by a special portal system. It's secreted in pulses, and that pulse of gonadotropic-releasing hormone controls the relation of these two hormones, which are present in both sexes. FSH is follicle-stimulating hormone. Follicle-stimulating hormone stimulates the chambers in the gonad where you're actually going to have meiosis and gamete maturation, so that meiosis to produce those four products and the development of those into sperm in the male, follicle stimulating hormone. Luteinizing hormone is also released, and luteinizing hormone has a number of secondary effects, the maintenance of secondary sexual characteristics and effects on behavior during mating opportunities. The primary cause in males of the release of these hormones is the production of the primary male androgen testosterone. Luteinizing hormone is the main hormone that increases testosterone production, and testosterone production is the basis of male sexual and reproductive performance. It's our most important androgen. It stimulates spermatogenesis and the maturation of those stages of development from the haploid gamete into the fully motile sperm. It also has an effect on behavior. So it shows libido, which is the psychological term for sexual drive, and related behaviors are vary according to the testosterone content of the body. It has an overall stimulating effect on the body, and testosterone does accelerate protein synthesis, blood cell formation, muscle growth. You can kind of see the importance of those processes, and leading to a kind of a vigor that would improve a male's chances of being a successful mate. Testosterone and development. The testosterone, in its constant production in the male, programs the hypothalamus again to control the release of those three primary hormones, which support a number of other things besides spermatogenesis. Sexual behaviors, I would call that mate identification and mate seeking behaviors. The types, I don't know what to exactly call it, courting, identifying a potential mate and attempting to move toward Congress, and sexual drive, the persistence that the male shows. In a number of species, testosterone is high in an alpha male, and other males are somewhat suppressed in their behaviors by the absence of testosterone. We do not have that alpha male system, so basically all males have this, their natural complement of sexual drive. Also, I want to make a distinction here about sexual characteristics, because by and large, we identify the sex of an individual based on their outward bodily appearance. But I want to kind of break this down. A male is defined by the fact that the male's body makes sperm. That's the primary sexual characteristic. That they make the motile and numerous gametes in this scenario, and that they deposit those gametes into the body of the female. The male is not equipped to receive the fertilized egg. It is not equipped to nurture that egg to live birth or to produce milk with the body after birth. The secondary sexual characteristics are all the other things associated with what we define as being male. If you think about it, the development of facial hair is one of the things that is distinct about the male. Muscle mass and body size typically are larger from a longer growth period, and that's primarily a testosterone effect. Characteristic adipose tissue deposits that are distinct from those in females are also male characteristics. In the growth of the male, the pelvic girdle remains more narrow and becomes taller and more robust to accommodate the higher body size and body weight. The shoulders in male typically tend to broaden. They grow wider to accommodate more muscle development. Those are all secondary sexual characteristics. The testosterone also maintains the accessory glands that we mentioned, the organs of the male reproductive tract. So essential to successful reproduction is to take that stream of sperm and turn it into semen. That's what makes it viable in the female reproductive tract, and that's a secondary sexual characteristic. Now, the male system is, this is all kind of tied into our, not so much our science, but our social discourse about gender and reproduction and sex. It's something that's very interesting. And so it's something that we talk a lot about. We represent it in writing and in videos and in stories. Often the classical stories, if you look at Greek and Roman myths, involve an element of sexual interaction among the main players. The distinctness of the male role, though, is that the male delivers a gamete, a sperm, and at that point, biologically, their contribution to conception and production of an offspring is done. Now, I don't want to in any way diminish our sort of cultural, societal, or religious traditions about the role of a male in a two-parent family, the statistics in sociology that examine the prospects of development of intelligence or success of offspring that come from two-parent versus one-parent household. That's all very important social information. But I'm trying to restrict this to the biological process alone. And so the male's role is to deliver that sperm. The female reproductive system is something that involves a much longer commitment and a lifelong commitment to production of high quality gametes, and then with each conception a longer commitment to the production of typically one offspring. We use this difference quite reasonably and quite importantly when we are applying it to sexually reproducing animals. So that a real fact, for example, of breeding cattle, is that once when you impregnate a cow, that cow is essentially tied up with that one calf, or more frequently than humans, twin calves. But it will take, I think 10 months is the mean for cattle. It'll take 10 months of that cow's life to come forth with one calf, and then that calf will nurse for a long period of time. So the frequency with which a cow can reproduce, typically this is managed on a year-by-year basis with one mating per year. Whereas the male, the bull, when the bull accomplishes fertilization with one cow, basically it can walk across the lot and accomplish fertilization with another. And as a result, in breeding stock animals, it is more valuable to have the bull because of the number of fertilizations that can be accomplished. If that bull is of high quality, then it will be much sought after as a mate. It's true in this, a parallel exists in the human female. From the point of conception, the female's body is tied up for 9 months in bringing forth typically one offspring. And that individual then will typically engage in an extended period of newborn care that involves nourishment, teaching, and protection. The high quality aspect is supported by the idea that each 28-day cycle typically releases a mature egg that can be fertilized. That high quality egg is also very fragile. I just heard a video the other day where the physiologist was saying 24 hours is really what they consider the viable period for an egg that has just been released, just gone through ovulation. The organs begin in the ovaries where the eggs are produced. They are released and move through uterine tubes to a muscular bag called the uterus. The uterus is, in a female who is not pregnant, is a small muscular sac that is fairly compact, lies behind and just superior to the bladder, and is connected through the uterine tubes to ovaries. Below the uterus, at the lower end, there is an opening called the cervix that leads to a passageway called the vagina. The vagina is that passageway that connects the uterus to the outside and the outside external genitalia of the female. So we are familiar with this already because we've looked at that urinary system. We've cut it off a little bit, but here's that bladder and the urethra opening here in the labial fold. Right behind it, superior, is the uterus, this kind of C-shaped muscular bag in the opening that we call the cervix. This is continuous with this tube, the uterine tube, leading back to the surface of the ovary. Now there's quite a bit of tissue here, and it needs support by ligaments and other connective tissue in order to suspend it here in the pelvic cavity. But this is the basic pathway for the egg. I'm going to kind of point out an irony about this, given that this is such an important point in human reproduction. This is the high quality gamete, but it's basically released from a follicle that matures, and it's basically popped out here into the space of the body cavity. Now this tube has finger-like projections surrounding its open end, and has the ability to move somewhat over the surface of the ovary and is attracted by chemical signals. But this isn't a hard connection. This is basically like a vacuum cleaner. There's a positive current flow toward the uterus, and in most cases the egg, when it's released, is vacuumed up and sent bouncing along this tube into the uterus. Now, where does fertilization occur? Well, it can occur just about anywhere, but what is ideal is if there is sexual congress and ejaculation into the tract in time for the sperm to make this long swimming journey all the way up here, basically to the end of the tube, and be waiting when ovulation occurs. That's ideal for fertilization. And so one way or another, either the unfertilized egg or the fertilized egg will be bouncing down this tube into the uterus, and fertilized versus unfertilized will then produce very, very different consequences of arrival in the uterus. Structural support: the ovaries, uterine tubes, and uterus are enclosed in a large sheet of dense, regular connective tissue called the broad ligament. There's plenty of structure around, since these organs are basically surrounded by the pelvis. So connection to the pelvis and to the sacrum and perhaps the base of the lumbar vertebra will provide plenty of support so that this broad ligament can support the weight initially of the female reproductive organs. The uterine tubes run on this broad ligament and open into the pelvic cavity lateral to the ovaries. The mesovarium is also another tissue that stabilizes the position of the ovary, because you're going to have two, a left and a right. They form a kind of a Y shape above the top of the uterus. Now we're seeing about half of this. Here's the vagina below and the cervix. This cervix is important. There has to be an opening for the sperm to get into the upper part of the reproductive tract, and after the baby develops, it's this cervix that will that will dilate and allow the passage of the baby down through the vaginal tract and out of the body. But the cervix is really important as a kind of a muscular ring. During the development up here of the baby, this cervix and its contraction, the competence of the cervix, is what holds the baby in its developing sac of fluid inside the uterus and supports it until its development is complete. So here we have the broad ligament holding these in relative positions. You think about this, we just look at this casually. These are tubes within tubes within tubes, And in order to function, they have to be open and allowing the passage of currents and cells up and down. So this is a great place to stop and to start next time, which will be next week after the test. So I'm going to stop the recording.